In this section, we are going to discuss steps of polymerase chain reaction. So, the first step is denaturing deoxyribonucleic acid into separate strands. The sample is heated to approximately 95 degrees Celsius for 1 minute to separate the deoxyribonucleic acid into single strands. Now, moving towards second step of polymerase chain reaction, which is annealing primers to flanking regions of single strands deoxyribonucleic acid. The sample is cooled to approximately 50 degrees Celsius for 45 seconds, as we have discussed. After denaturation, single strands deoxyribonucleic acid will be formed. These are flanking regions and the 2 primers, one for each strand. Anneal to a complementary sequence on the single strands deoxyribonucleic acid. Moving towards third step, and that's extending primers with deoxyribonucleic acid polymerase. The sample temperature is raised to approximately 72 degrees Celsius for 2 minutes to initiate the synthesis of 2 new strands complementary to the original deoxyribonucleic acid strands. Deoxyribonucleic acid polymerase adds nucleotides to the 3' hydroxyl end of the primer, and strand growth extends in the 5' to 3' direction across the target deoxyribonucleic acid, making complementary copies of the target by using a heat-stable deoxyribonucleic acid polymerase, such as TAC from the bacterium Vermis aquaticus, that normally lives at high temperatures. The polymerase is not denatured, and therefore does not have to be added at each successive cycle. TAC deoxyribonucleic acid polymerase is heat resistant, so it is not necessary to freshly add this enzyme for each cycle of polymerase chain reaction. However, TAC lacks proofreading activity. Typically, 20 to 30 cycles are run during this process, amplifying the deoxyribonucleic acid by 1,000,000-fold to 1,000,000,000-fold. The polymerase chain reaction products from this extension can be several thousand base pairs long. At the completion of first cycle of replication, 2 new double-stranded deoxyribonucleic acid molecules will be formed. Now moving towards second cycle. The reaction mixture is heated again. Means denature deoxyribonucleic acid into separate strands produced from first cycle. So, heated as 95 degrees Celsius to separate the strands, of which there are now 4. Next step is annealing. So, each strand anneals to the complementary primer. Third step is extension. So, the step of primer extension is repeated. Now let's summarize what we have discussed in this section. Well, you can remember the requirements needed and steps for polymerase chain reaction, both by 3, means 3 needs, and 3 steps are required in polymerase chain reaction. So, let's name the needs, which are flanking regions, primers, and deoxyribonucleic acid polymerase. Now, let's name the 3 main steps required for polymerase chain reaction, which are denature deoxyribonucleic acid into separate strands. Second step is anneal primers to flanking regions of separate strands. And third is extend primers with deoxyribonucleic acid polymerase. But before ending this section, let me ask a question from you. What decides the temperature for denaturation of deoxyribonucleic acid?
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.